Module 2, Quantitative and Population Genetics in Livestock. Module Overview. Module 2 delves into the principles of quantitative and population genetics, providing a robust framework for understanding how complex traits in livestock are inherited and how they can be manipulated through scientific breeding strategies. Unlike simple Mendelian traits controlled by a single gene, many economically important traits in livestock such as milk yield, growth rate, feed efficiency, fertility, and disease resistance are polygenic, influenced by multiple genes interacting with environmental factors. This module equips you with the knowledge and tools to analyze, predict, and improve such traits across populations. The module begins by defining quantitative traits and their continuous variation in populations. Individuals explore the statistical concepts underlying quantitative genetics, including mean, variance, standard deviation, and coefficient of variation, which are crucial for interpreting performance data in herds and flocks. The module emphasizes the partitioning of phenotypic variance into genetic and environmental components, providing insights into how traits can be selected for improvement. A major focus of Module 2 is on heritability and repeatability, explaining how these measures quantify the proportion of phenotypic variation that is due to genetic factors. You examine narrow-sense heritability, additive genetic effects, and broad-sense heritability, total genetic effects including dominance and epistasis, as well as their implications for selection efficiency and breeding program design. The module also covers genetic correlations, demonstrating how selection for one trait can affect correlated traits, either positively or negatively. For example, selection for rapid growth in beef cattle might influence feed conversion efficiency or reproductive performance. Individuals study selection differentials, response to selection, and selection intensity, which are essential for predicting genetic gain and designing effective breeding programs. At the population genetics level, this module introduces you to concepts such as allele and genotype frequencies, Hardy-Weinberg equilibrium, genetic drift, inbreeding, and effective population size. These concepts provide the foundation for understanding genetic diversity, population structure, and the long-term sustainability of breeding programs. Inbreeding depression, caused by mating-related individuals, is discussed in detail, emphasizing its impact on fertility, growth, and disease susceptibility. Furthermore, Module 2 integrates modern approaches to population management, including pedigree analysis, estimation of breeding values, and selection indices. These tools allow breeders to make data-driven decisions for the genetic improvement of livestock populations. Molecular markers and genomic tools are introduced as advanced options to complement traditional quantitative approaches, enabling precise marker-assisted selection, MAS, and genomic selection for polygenic traits. By the end of Module 2, you will have a thorough understanding of how genetic and environmental factors interact to influence complex traits, how to measure and interpret these effects, and how to apply this knowledge to maximize genetic gain while maintaining population health and diversity. Key learning outcomes. After completing Module 2, you will be able to define quantitative traits and describe their importance in livestock production, explain phenotypic, genetic, and environmental variances and their contributions to trait expression, calculate and interpret heritability and repeatability for selection purposes, analyze genetic correlations and understand their implications for multi-trait selection, apply principles of population genetics including allele frequencies, Hardy-Weinberg equilibrium, and effective population size, identify and manage inbreeding, genetic drift, and selection intensity in livestock populations. Use selection indices and breeding value estimation to make informed genetic improvement decisions. Integrate molecular markers and genomic selection into population-level breeding programs.